Hello, everybody. Thank you so much for coming to our Art of Life Learning Community presentation. We're very excited to tell you about this very integrated course we taught in fall 2016. In a nutshell, this course was very much an out-of-the-box learning experience for our students. We turned everything upside down from the curriculum to the format, made it very project-based, and really made a unique experience for our students. My name is Bree Day. I'm a full-time faculty member in biology here at Eastfield, and I team taught this with Val Curry, who is a part-time faculty member in art. So how do you integrate art and biology? Well, it wasn't easy, but we had a lot of fun. So let's take a look at some of the details of how we set up this course, and more importantly, actual photographs of our students in action, which I think says a lot more than anything we could tell you. So we're calling this presentation The Art of Life, a Radical Experiment in Out-of-the-Box Teaching and Learning, because that's really what it was. It's a very experimental course, and I'm so grateful to the Eastfield administration for allowing us to attempt this experiment. I've been wanting to teach the Art of Life course for about 10 years at different institutions, and every time I would request it, people would look at us and go, well, how are you going to meet course objectives or outcomes? You might not be able to cover all the curriculum. People were very skeptical of it. And so it wasn't until I got to Eastfield that I proposed this to my dean, and she said, go for it. Awesome, thank you so much. And then lo and behold, as we were talking about learning communities, suddenly Val Curry sent me an email and was interested in teaching this, and I'll let him talk more about that. So, and he had the same vision that life is beautiful, life is art. And we both were very interested in bringing some of the beauty that nature provides to our students in an artistic fashion. So this is a team-taught course, truly integrated. A lot of learning communities are what they call co-taught, where each instructor in different disciplines teaches their own course as they normally would, but they have assignments or projects that have a common theme. And so they try to link the curriculum to give the students more of an idea of how different disciplines can be integrated. With team-taught learning communities, which is what we did, it's different. It's both instructors are in the classroom at the exact same time. We're both interacting at the same time with the students, and all of our projects and assignments are completely integrated, meaning instead of doing an art project and a biology project, we do a bio-art project, or an art-bio project, if you will. So this took a lot of guts to experiment, and a lot of it was done as we went along, but in the end the students really loved it and they got a lot out of it. So let's find out some of the more details of what the features were of this Art of Life team-taught learning community. So the students in our Art of Life learning community were all non-majors. They were neither biology majors nor art majors. They get 7 credits for coming to campus only 1 day a week. And now how in the world do they do that? Well, you've probably heard of the flipped classroom, and this really was kind of a flipped classroom where instead of, for example, in a typical non-majors biology class, the lecture is kind of the crux of the class, and the lab is meant to supplement the lecture. We did this the opposite. We made the lab, the project-based part of the course, the feature part of the course, and that's happened on the 1 day they came to class. And the lecture was meant to supplement the lab. And we did that through the internet. We made this a hybrid online course using Blackboard. And I'll show you a little glimpse of kind of how the setup was for that in a second. So 7 credits, only coming to class 1 day a week for 4 hours. They actually come for an hour discussion session before the lab where we can review the concepts they learned in the online component of the course. And we also really had very interesting discussions. It was a very discussion-based hour where we would talk about controversial topics in both biology and art, talk about people in the field that are integrating those two fields. It was really, really fun and really got people to kind of think critically about certain subjects. A lot of them controversial things like genetically modified foods or organ printing on 3D printers, things like that. And then they would go to lab for 3 hours where we would do completely integrated lab-based and project-based projects that blended art and biology. So very student-centered in the sense that a lot of students have trouble getting 7 credits out of the way, and this allows them to do it in a very flexible kind of schedule. So again, both instructors are present in the classroom. both in the face-to-face and in the online environment. Our projects were fully integrated, and the course was project-based. So not only did we flip the format of the class by making the focus the lab and the supplement the online lecture, but we also flipped the curricula. And this is for both biology and art. In other words, if, for example, I can speak to biology, I'll let Val speak to art, but in biology, you know, the traditional way is you start with chapter 1, and then you go to chapter 2, etc. And it usually starts with the scientific method, and then you go over some basic chemistry and parts of the cell, and you build up over the course of the term, and right at the end you start talking about DNA and a little bit of biotechnology. Well, this was flipped. We started with day 1 with DNA, the molecule of life, and we started right on day 1 with the students extracting their DNA from their cheek cells. They started using the microscope the very first day, and they also were using some techniques in biotechnology. And that theme of DNA and biotechnology and the central dogma of biology was carried through the entire course. In fact, they didn't start learning about the other parts of the cell, really, until right towards the end of the course. So completely flipped curriculum, and we found that actually really made students more engaged. They got interested because they were doing topics that were related to their lives as non-majors, and it was really fun. And I'll let Val speak to how he flipped the art part of it. So students got credit for both Biology 1408, which is the first semester non-majors biology course. It's a 4-credit course. And they also got credit for art appreciation. That's Arts 1301. So both of those courses required for their programs, and boom, got those out of the way. Now, one of the things we found, and in doing our research, we found this to be true, is that by making project-based courses that are student-driven, that are team-taught and integrated, and where the curriculum is flipped, it tends to activate multiple parts of their brain. So, you know, the science side tends to be one part of the brain, and the more art creative side tends to be another. And by integrating these two disciplines together and all the projects involved with them, it actually activates multiple parts of the brain. It really gets students thinking outside of the box and thinking about things in a different format. And we totally found this to be true, and you'll be able to see this in the pictures of the students that we show you. We also worked on, focused on collaborative learning. So almost all of the projects we did in the course and many components of the online course were collaborative. They were team-oriented projects. We had collaborative discussions in the face-to-face discussion section, as well as in the online learning environment. We had interactive discussion posts about controversial topics related to biology and art, and the students were required to respond to our post to them, as well as at least 2 classmates. And then in the discussion sections, we got really, you know, we'd almost have debates going on. And what's cool about having a team-taught environment is even the 2 instructors don't always agree on things. Like, we had a very interesting conversation about vaccines, for example, one day. And they can see that, you know, even academics, you know, even though we've been through all these years of schooling and graduate school, we have different perspectives and different takes on topics. And I think that's a really valuable life lesson, college lesson for students to understand, is that you can have multiple perspectives on a topic, and it doesn't mean that one's wrong and one's right. So the other thing is, in addition to just the format of having a hybrid course where they're only coming to school 1 day a week, we really tried to make this course student-centered. In other words, the students helped to take charge of their own learning. We provided a basic framework and the basic projects that were going to be done, but we really kind of, especially as the semester went on, we sort of took the reins off and let them decide which direction they wanted their learning to go. For example, at the end, we'll show you one of their really off-the-wall art projects we had them do, where we basically said, here are the minimum requirements. Now you guys work as a team, you figure out how you're going to solve this particular problem, and go for it. And we just sat back and watched, and it was just absolutely remarkable, and we'll get to that a little bit later in the presentation. So students really did help to direct the curriculum and the projects. It was really, really fun to watch, and over the course of the term to watch them grow in their ability to do that. The other great thing about having a truly integrated team-taught learning community such as the Art of Life is we're able to very easily target multiple learning styles. You know, some students are better at reading and listening. Many students are better at visual learning and tactile learning and social learning. So by having such an integrated course, we're able to address all those different types of learning styles kind of all at once. And so you really get buy-in and integration by all students because there's something for everybody within a course like this. And again, we really tried to focus on things that had real world learning and application. So things in biotechnology that may be affecting their life or their kids' lives someday, such as, you know, what kind of food are you going to buy? Does it matter if it's genetically modified or if it's organic or not? You know, how do you view stem cells and cloning, and you know, these kinds of controversial issues that come up in everyday life but are based in science, and then for them to appreciate how things they see in their everyday life are beautiful that nature provides. In flipping the curriculum, one of the things we were really focused on as well is making them understand that beauty provided by nature and modified by humans, it can happen at all levels, even levels that you can't see with your naked eye. So in this course, they not only did a lot of macroscopic stuff, but they also learned to look way, way deep using a regular compound microscope, a dissecting scope, and even the electron microscope that we have here at Eastfield. So they're really seeing how beautiful everything is at all levels, and then they learn to capture that beauty in their own art projects. So this is just a screenshot of our welcome page in our online part of the course. And just to show you that we even tried to integrate some artsy kind of stuff, you know, with these fancy backgrounds to Blackboard within the course. Right when they come in, they get a welcome letter from Val and I, and Val and I created a little welcome video to welcome there and tell them about the course. It took us 5 hours to put together because we had to keep restarting it, but it was a lot of fun. We wanted to kind of bring our personalities into the online environment since they were seeing the online component before they actually saw us in a face-to-face class. But again, as you see, kind of our format is as few buttons as possible over here, really focused on the discussion board, which we called the Art of Life Chat Cafe. And with the learning units, we actually would have a folder for, okay, here's the biology stuff, here's the art stuff, and here's where it's integrated, so that they would get a nice background of the actual, you know, content, course content, making sure all the curriculum and the objectives are covered. And then when they come to class on Fridays, then that's when we bring those concepts alive in a project-based format. So, you know, we had a lot of self-filmed lectures as well as lectures from other people. So we try to do a lot of video-based lectures. For the biology part, I used modified mastering biology. So the students were still doing the exact same homework assignments and online exam questions that my normal online traditional 1408 would be doing. And then the tests also integrated the art questions over the curriculum. So they're still being assessed in the traditional format in an online setting over the course content. And then the real assessment to us was the projects that they actually created on Fridays. Okay, enough basic background. Let's get into the good stuff where you actually get to see the projects that the students were doing. What I've done for each of these, so we had 7 basic projects, and some of these were multiple weeks, some of these were done in one day. And what I've done is, before I show you the actual photographs of the students, which is my favorite part, we've actually just outlined what biology objectives or outcomes and skills were included with each project and which art outcomes and objectives and skills. Now you have handouts you know, of what you're seeing here, so I'm not going to go over each detail, but just kind of sum up. So starting with the first project, this is week 1, day 1. We had the students do a lab activity that's actually a kit that they get from a company called BioRad, which is totally rad. And it's called Genes in the Bottle, cheek cell DNA extraction. So the students used this kit. They literally extracted their DNA from their cheek cells. They could actually see it. It becomes a physical, you know, macroscopic thing they can see. They had a little necklace kit, so they actually put their DNA, their actual pure DNA, into the necklace. And that way, they can give their DNA to their loved one or keep it for themselves. And then to kind of hit home and sort of integrate this idea of the basic structure of a cell and how you can fit all this DNA into the little nucleus of a cell. After they've extracted their DNA, the students actually learned how to use a compound microscope, and these guys actually used oil immersion, which normally we don't let our non-majors do, but these guys, this is a good group. So 11 students, and we could work really closely with them. They learned how to use the microscope, and so they created wet mounts with their cheek cells like they would in a traditional biology class, and they learned to stain it. They looked at their cheek cells at different magnifications, so they could get the concept of, okay, that DNA came from this little thing that I just pulled out of my mouth. And then Val taught them how to do what are called cheek cell grid portraits. And so they actually had to take photographs of their cheek cells through the oculars of the microscope and then use the techniques in grid portrait to create beautiful painted canvases of their cheek cells. And you'll be able to see some of the remarkable products they came out with this. So you can just check out, they're learning a bunch of different skills that they might learn over the course of several weeks in a traditional biology class. They're doing it all in one day on the first day of class. And while they're at it, they're beginning their study of the structure of DNA and DNA's discovery, which will carry over for a couple weeks, including the idea of women in science. So there's some ethical issues there. They talk about Rosalind Franklin and her role in the discovery of DNA. And then for art, I'll let Val quickly go over all of these different skills that they would normally learn in an art appreciation class, or at least develop an appreciation for, and they're actually applying these things in this class. So ready? Here we go. So here are some photographs of our students and our Val Curry here with their extracted DNA made into necklaces. These are actual pictures that students took of their cheek cells at various magnifications. So you can see that they're taking it with their cell phones actually through the ocular of the microscope. It's a little tricky, but they did it. And then their beautiful cheek cell grid portraits that they ended up with at the end. And so for example, here's Jessica, and you see there's her cheek cell portrait, and here's her actual cheek cell. So she did a very good job of capturing what she saw in the microscope onto the canvas. And again, these are non-majors, so they didn't come in being artists. Val Curry has brought out their inner artist in a very fun way. And as you can see, the students are just having a blast with this. So it was a really great way to start off the course hands-on and still give them that fundamental understanding of science and art concepts. Okay, week 2, DNA paper craft and origami. So again, DNA is a theme of the entire course, that life is beautiful and it's based, no matter whether you're a human or an elephant or a tree or a virus, it's based in this molecule, DNA, which is a simple molecule, yet elegant. And so we really wanted them to understand the structure of DNA, both the molecular structure as well as the beauty that's in the shape of it being a double helix. And so the second week, we actually kind of got away from the microscope and had them do what's called paper craft or origami. So this is very much an art technique, and you can see what all these concepts that they're learning in art. And so biology, we're carrying over, understanding fundamentally the DNA structure and function at the chemical level and its discovery. So continuing to talk about Rosalind Franklin and they watched a movie called The Secret of Photo 51, which is about how some of the ethical issues surrounding the discovery of DNA and how Watson and Crick, who are credited through their DNA model, got the Nobel Prize, and Rosalind Franklin, that they actually stole her photograph of her X-ray crystallography, and she ended up dying early, got really very little, if any, credit for her discovery. So we talk about those issues of women in science and what it was like to be a woman scientist back in the 50s. And I actually, in the online environment, they actually read the actual science publication of when Watson and Crick published their discovery of the structure of DNA. So it really, makes for a fun discussion section, before the lab, where we're talking about the ethics behind the discovery of DNA. And then for this actual lab, Val taught them how to fold paper into structures that were the actual shapes of the molecules that make up DNA. And so then they build it, and here we see what they have here. So this is our class actually building this model. One of the things they learned is that you can fit about 7.69 feet of DNA, if you were to line it up end to end, into the nucleus of one cell. So the previous week they extracted their cheek cells and they saw the nucleus under the microscope. They understood about magnification and what the actual size of the thing is. And then they actually get to ponder how much DNA is in one of those cells. So here's Val building a 7.69 foot pole that they're going to build their DNA origami model on. And so they can see the actual size of this thing and think, wow, that's in the nucleus of one of my cells that I just looked at under the microscope last week. So it really puts things in perspective for them. And then here are some of our students. Here's Larry drilling holes into the pole because the students actually took skewers and they here are the actual origami shapes that reflect the actual shapes of the molecules that make it up. And Val was able to actually figure out how to place the holes so that it rotates at an accurate double helix. So anyway, you can see it was a lot of fun. It was collaborative. We only got a third of the molecule built this semester. And so what we're planning is over the course of next year and the year after that, by the end of 3 years, we will have the entire model built. But it is art. It's beautiful. It shows how intricate and beautiful this model is. And you can see in the background here is Watson and Crick with their actual model that they built to examine the structure of DNA. So it kind of puts the historical context of DNA into the project as well. Okay, week 3, we carried their understanding of DNA and now put it into application in terms of forensic DNA fingerprinting. And so we once again used a Biorad kit, and this one they're actually using real DNA, and they're using what are called restriction enzymes that allow them to cut the DNA at certain points. And they're kind of playing CSI here, where they're learning to understand how, for example, at a crime scene, they would take DNA samples from the crime scene and DNA samples from the suspects and use biotechnology to figure out, what's the likelihood that this person committed the crime. And we also talk about the ethics around DNA fingerprinting and whether, just because you have a match in a DNA profile, does that mean the person actually did it? So anyway, and then we carried that over into the art aspect of, wow, a DNA profile where you see the little bands, everybody's probably seen it on TV, is actually beautiful. It itself is art. And so Val taught them a technique called printmaking using a glow-in-the-dark paint. And the students actually did screen printing to make their own t-shirts. We had them bring in blank t-shirts, and they actually screen printed a DNA profile that glows in the dark onto their t-shirts, and they got to take that home. And so we'll show you pictures of those. But this was actually a very biotechnology rich lab. So they actually learned all these things that most of even our majors don't get to learn. Things like gel electrophoresis. So they actually got to make gels and use real DNA in there and use restriction enzymes and run those gels. And I'll show you pictures of it. And, a lot of times people think that maybe non-major students or even major students might not be able to do such complicated work. But we proved them wrong, and you'll see these students learn mechanical pipetting, and incorporated in all this is the scientific method and experimental design. So even though we don't have a lab just on the scientific method and measurement, they're learning that organically through our discussions and through the actual processes of doing these labs. So check out some of these amazing pictures. So well, this one might kind of be weird. What we actually did to help them understand the context of DNA fingerprinting in applied things like crime solving, we had some wonderful volunteers. You can see here Dr. Reel and Amy Vance and a volunteer student that I grabbed out of the hallway. These guys represented the suspects because we actually turned out the lights, and Morgan, who's not with us anymore, dressed up in a cloak, and we turned out the lights, and she came and murdered Val here. So here's Val Curry, dead in the middle of the lab. and so we staged this crime scene, and then we brought in our suspects here. We had 5 of them, and we showed the students how, we had someone pretend to be the investigator, and they came and took DNA samples from their cheek cells. And then we said, okay, so now you guys, the class, are the forensic scientists who are going to go and actually extract the DNA from these samples and run them on these electrophoresis gels so that we can compare the DNA from the crime scene around Val here to the DNA of the suspects and figure out who killed Val. Val was a very good actor for this, by the way. So we brought them, after our little skit, we brought them downstairs to what we call the biotechnology lab, the instrument lab, where we have all sorts of amazing biotech equipment, thanks to Danita, who wrote a grant to acquire this equipment. We're a very lucky school to have this kind of stuff. And she was nice enough to let us use this equipment. And so we taught them basic, micropipetting techniques. This is a great skill. All of them can now put this on their resume. People can get hired in industry as lab techs just because they know how to pipette. So this was a very, very good skill for them to learn. They learned techniques of what a microliter is, and very good stuff. And then you can see that they're actually, creating their own electrophoresis agarose gels. They're loading the DNA into them. They're using all sorts of biotech equipment in the process, and you can see from their faces they are extremely engaged in this. A lot of them reported they feel like real scientists. So again, these are non-majors, but really just completely got engaged in this process and learned so many different topics in biology while they did this. And so here's some more shots of them having actually loaded their gels with the DNA and actually running them. They're using microcentrifuges. I mean, it was just so much fun and this is only the third week of the class. In the process, they're measuring out powders using gram quantities. So they're learning a lot of different techniques that might be a focus of a lab in a traditional class. This is just incorporated into what they're doing. And then as they're waiting for their gels, they go upstairs to our upstairs lab. And this is where Val is teaching them how to do screen printing with this glow-in-the-dark paint. And so here are their DNA profile pictures that they are actually putting onto their own t-shirts. Val was a genius and actually was able to get Art of Life 2016 on the picture that he had them screen print. And you can see they're all engaged, they're all happy and having fun. They got to take home a souvenir, which always helps the students to think about what they did after they leave the classroom. And here's their glow-in-the-dark paint. And when you turn out the lights, it's really fun. It's just this glow-in-the-dark DNA profile. So they've gotten the science and the art integrated in the sense that they're thinking about this. They're like, okay, what is this thing I just put on my t-shirt? Well, it's the DNA profile, and they can explain to their friends and family when they get home what it means. So really helps to reinforce the concepts that we're trying to get them to learn. So this takes us to week 4, where we're now ready to branch out. They understand DNA and some of its applications, but how does it actually fit into a cell? And so we wanted them to gain an appreciation that 7.69 feet of DNA is compacted and wound so tightly into the nucleus of each cell that it can all fit there. And 30,000 genes that make us who we are all fit into the nucleus of a cell. And so we wanted to make them understand the beauty of that in an artistic fashion. And so we actually taught them, Val taught them the techniques of molding and casting and 3D printing and sculpture, as well as edible art. And I taught them about how that DNA is packaged into chromosomes by wrapping around little proteins called histones. Now, unfortunately, this happened to be the day when the power went out on campus. And so our lab was cut very, very short as a result, and we weren't able to spend as much time on this as we wanted to, but they still were able to get the basic concepts, and they did actually mold both edible chromosomes as well as plastic chromosomes. And Val had 3D printed a chromosome at UT Dallas, and so he was able to explain to them the process that he went through to create that 3D sculpted mold and what some of its applications would be in art. In the process, they're also learning about karyotyping, which will carry over into a future lab that we'll talk about in a minute. So here are the students doing their molding. Here's Val mixing up the two mold materials. And here's their little chromosome that they're pouring. So that's the non-edible one. This is the edible one. They actually used pink chocolate melties and fruit roll-up things and gumballs. And so the gumballs represented the histone proteins. Here's their edible chromatin, and here's the actual chromosome that these tighten up to wind into the chromosome here. And we have the actual model of a chromosome that's just sitting there in the lab, so they can kind of see how these things wind up into the finished product. So very fun. They still had to use skills like weighing things and kind of putting it all together. So it was brief because of the circumstances of the campus getting closed, but it was fun, and they, as you can see, they seem very engaged. So once they understood the structure of DNA, its applications, they learned DNA replication, so how DNA replicates itself, and how it's packaged into chromosomes. They are now ready to dive into one of the hardest concepts for a non-majors biology class to tackle, and that is the central dogma of biology, aka protein synthesis. How do we go from the genes on a DNA molecule to a protein to the actual trait? It's a very complicated process. We don't go over all the details that we would in a majors class, but we thought a good way for them to understand this concept is to be able to focus on it by doing a project that's very integrated in terms of how much time they have to spend on each component. And so Val taught them how to create video clay animations. So if you've ever seen Wallace and Gromit and the clay animation, how they, do a little thing, they stop the camera, they change the clay, start it again, and it ends up becoming an animated cartoon where the characters are made out of clay. So he taught them how to do this kind of video animation and stop animation? Val, you'll have to help me on that one, as well as sculpture and storytelling. And so they were tasked with taking either just transcription or translation or both, which are part of these processes involved in protein synthesis, and creating a clay animation video showing this process. So they're learning protein synthesis because what they have to do is stop and start their video so many times to make the clay animation that it really makes them stop and think about what's going on in the process. Of course, they're learning all these other concepts at the same time: molecular evolution, bioengineering, that kind of stuff. So we thought what we would do for this is show you one of an example clay animation video that one of our students made. So here you can see the students actually working on sculpting and sculpting the clay. These are just screenshots of the YouTube videos created by our students, but we're going to actually show you this one by our student Taylor. She did transcription, and she did a very good job of showing the process all the way down to the details of how different nucleotide bases are stuck to the end and the beginning of the mRNA transcript, and how the introns are spliced out before the mRNA transcript goes to the cytoplasm. If that sounds like a foreign language, don't worry, at least our student learned it. So we're going to show you that real quick. I'm going to let Val start that. The next week, we really get to put a lot of their techniques they've already learned, from basic biotech kinds of stuff like pipetting to creating videos. They're sort of compounding these skills together now into what was one of my favorite projects that they did. This is really fun. So from Biorad, we used a kit called the PGLO Bacterial Transformation Kit. And in this kit, it's so neat. They actually, they take bacteria and harmless bacteria, and they actually insert, using biotechnology techniques, they actually insert the glowing jellyfish gene, the gene that makes certain species of jellyfish glow, bioluminesce, and they actually insert it to the bacteria along with a gene for antibiotic resistance. And they grow up these bacteria, and the ones that were transformed with the antibiotic resistant gene along with the glowing gene will glow in the dark. And so it really helps the students to understand protein synthesis, so how putting a certain gene in changes a protein, like the glowing protein, and causes a trait to happen. And in the process, they're learning microbiology techniques like the aseptic technique and how to grow colonies of bacteria. They're understanding prokaryotic cell structure and how it's different than eukaryotic cell structure. They're learning good lab safety, scientific method, and experimental design, because they have control plates and different treatment groups. So they're understanding all that. Molecular evolution, ethics and bioengineering. I mean, you name it, they are learning so much in this one lab, and it's a cool lab, it's fun. And so this is a 2-week lab. Once they have actually transformed the bacteria and they get some of that glow, they are actually going to do more video art but this time we're also incorporating things like social commentary, because the idea is, what are the ethics behind bioengineering and transforming organisms to something that they naturally wouldn't be? Maybe how do you feel about that in terms of just labeling bacteria for antibiotic resistance versus how do you feel about making a monkey glow in the dark, or making genetically modified foods, et cetera. So we get into all these ethical discussions that we discussed in the online environment and in our face-to-face discussion section. And then their art project for this is they had to create a video art where they start with the GFP protein, that's the green fluorescent protein that's extracted from the jellyfish. And so they see the structure of that, and then they were required to go from the bacteria that they actually grew. They took pictures of their own glowing bacteria, and from there go through as many different organisms as they could locate on the internet that had been made to glow using that same protein, the green fluorescent protein. And at the end, they're supposed to put sort of a question mark as to, how far should we let this go? Should we be able to transform humans like this? And so we kind of took the reins off and let them be creative in how they wanted to present that social commentary. But the idea is that it is an art form to include social commentary into the actual art. So we call this the secret of life presented in form, light, and motion. So here are the products of their work on plasmid transformation. So you can see the plates that had the transformed bacteria with the glowing jellyfish gene that the students inserted glowed in the dark. We turned the lights out in the lab, and they used a UV light, and they were able to actually see their bacteria glow in the dark. It is so cool. And then here are the plates that don't glow. So they got the idea that, okay, well, those did not receive the antibiotic resistant gene along with the GFP protein. So you can see the students, it's a little dark and grainy, but you can see they're having a great time. Amy Vance, thank you, Amy, actually had them do the first week of this because I was out having surgery. And so she, in the discussion section, actually had them create these little paper plasmids. So they were kind of doing an art project to prep this and to help them understand how a bacterial chromosome is set up and where these genes are inserted and that kind of stuff. So that was really awesome. So once they've grown this up in the lab, then they actually create their video art. And so all these different organisms, from a bunny to a cat to you name it, have been made to glow in the dark under a UV light with this protein, the green fluorescent protein that is extracted from jellyfish. And so these are screenshots of our students' different video art projects. We have them put them all on YouTube, as well as share them with each other in the online discussion board. This one's actually interesting because this bunny is actually a transformed bunny, but was made by an artist onto canvas as a piece of art. So that was already done, where someone had pondered, are these genetically transformed organisms art? And so we raised that question. So I'm going to let Val play Jessica's P-Glow video here. She does a really cool job, and it's a little bit long. It's about 3 minutes, but at the end, you can see her social commentary she threw in there. She did a very good, very creative job. So I think you can see as time goes on, the students are building their skills, building their ability to integrate biology and art together and think kind of outside of the box. And so the next week, we really put their skills to the test. We had them do a kit from Biorad called GMO Investigator, where they're actually doing PCR, polymerase chain reaction. And that's where they actually isolate a gene, a particular gene from DNA from an organism, and they use a PCR machine, also called a thermal cycler, which we are so fortunate to have. We're one of the few community colleges, I would say, that has one. And they actually use that to make billions of copies of that gene, which they then run on an electrophoresis gel, which they learned early on in the semester. And from that, they can determine the presence of that gene in a sample. And so what we actually had them do is bring in their own food. So they could bring in organic food, or they could bring in processed food that they suspected to be GMO. And we were looking for the presence of one or both genes that are present in about 80% of the genetically modified crops in this country. And so if their food was genetically modified, those two genes would, one or both of those genes would show up. So they're back in the biotech lab doing all the biotechy stuff, so same thing, jello electrophoresis, mechanical pipetting, metric measurements, etc. They're making solutions, so they're learning all that stuff. But they're learning PCR, which is a fairly complicated process. You're dealing with microliter quantities, and you have to work over ice the entire time. So it's easy to make a mistake and have your results not work. But they were up for the challenge. They did it. Hey, if our non-majors can do it, anybody can do it. And then after that, because one of the things about PCR is there's a lot of waiting around time. It actually takes 4 hours to run a sample on the machine we have. And so during the first part of their waiting period, we actually had Val teach them drawing skills. So that was still an art skill we wanted them to develop an appreciation for. And so he taught them all these skills for drawing, and then they actually drew the PCR machine that they were running their samples on, the thermal cycler. And so here they are. So here's the actual biotechy part. So you can see they're back to loading gels, but here they're actually making up their samples. This was right near Halloween, and normally you'd get coolers filled with ice for them to work over, because you have to work over ice so the DNA doesn't denature. But I couldn't find those at the grocery store. Instead, I found dollar Halloween buckets. And so we use those, and we're still actually using them to this day because they actually work really great. So the students by now had already developed the basic biotech skills they needed to do this more complicated lab. This is the actual PCR machine, the thermal cycler. And once they run this thing, it's basically a glorified oven. And as they're running it, they're going to draw it. And so one of the things Val had them do was read this article called Rediscovering the Forgotten Benefits of Drawing. And it's really neat. It's actually written by a biologist at a University of North Carolina, where he's focused his biology labs on bringing back the art of drawing. And so everything they look at under a microscope or in the lab, they're learning to draw using really sophisticated drawing techniques. And so that was a really good introduction for them to read. And then here are some of our students' drawing projects where they're drawing this thermal cycler machine. And we think they did a great job, and we featured some of their artwork in our gallery, which was very cool. They were actually part of an art show. Okay, now this is the other thing that was really interesting about this class is we just happened to be offering this class in the year where the common book of the term was The Immortal Life of Henrietta Lacks. And that book just was perfect for this class because it was very featured in terms of the biology, as well as, of course, the ethics of women and minorities in science, and actually had some art in there because they talked about creating a picture of Henrietta Lacks's chromosomes and giving it to her friend as an art piece. So it was really cool, and we wanted to really feature this common book in our course. So our students actually entered the common book competition on campus, and this is so cool. We actually, along with Pebble, Barbero, and Danita, we actually got hold of a kit put out by NIH where the students actually work with real HeLa cells. That's actual cancer cells descended from the same cells that were taken without her permission from Henrietta Lacks. So the students are actually working with her cells, and that really, it gives an awe. It's an emotional reaction that the students get because they know that they're working with the same cells they just read about. And it makes them think about cell replication, like mitosis, and how, cells divide, and how these cells that they're holding in their hands are descended from the same cells from back in the early 1950s. And so they really think about mitosis and cancer at a deep level. The actual lab, they're taking these cells and doing a very interesting technique where they drop them from 2 feet up, and that bursts them, and then they can actually look for chromosomal abnormalities that are common in cancer cells. They learn about karyotyping, pedigree, inheritance, and of course, involved in all of this, we're talking about the history and bioethics issues that were surrounding these HeLa cells and what the impact of these HeLa cells have been on science and medicine. And so from this, after they actually do this lab, They then are creating their art projects as teams that they're going to enter into the Common Book competition. And so they're learning these different concepts, including something called cultivating creativity that I'll let Val talk more about. But the idea is that, hands are off. We don't tell them what kind of art project to do, but we just say it has to be based on what they learned from reading the Common Book and from doing this HeLa Cell Lab, and that they have to incorporate skills they've learned in the art component of the course as well, and make an art project that they can then enter into the competition. And so they really did a great job. So here they are doing their HeLa Cell Lab, where they're actually dropping these cells from 2 feet up. So it was very engaging, very fun. And then here, of course, when the relatives of Henrietta Lacks actually came and spoke, that's really cool. So it kind of really just tied everything together, really made the students engaged. And when you look at their final projects, they just did a great job. So we ended up having 3 entries into the competition. So you can see one group really, here and this group as well, both highlighted cancer cells. So these are supposed to be cancer cells that were stained with the green fluorescent protein from the jellyfish. So they're actually kind of taking something they've already learned about and incorporating it into their art project and showing how that by itself is art. And then you can see in this one, they highlighted some of the amazing breakthroughs in science that have been accomplished as a result of using Henrietta Lacks' cells. So what her impact has been on the world of science and medicine. Okay, second to last project they had to do. This is the found object art of a cell. So this is the one where we're totally hands off. We let them just lead this project. All we told them was that they have to bring in objects or find objects on campus, and out of it they're going to create some kind of cell in a very abstract and sort of modern art way. And so they had to work as teams to create one project. So basically as a team, they had to work as an entire class on this one art project. They had to use cultivating activity and something called found object art. And so we had been talking in discussion and lecture about found object art. Like, for example, they learned about an art project where the artist took a urinal and turned it on its side and signed it, and that became a very famous art piece. And so we asked this question: what is art? Where do we draw the line of what's just putting something together and what is art? And so then they had to take those concepts and apply them to creating a project of a cell. Now these students decided they wanted to create a sperm cell, because we were also learning about meiosis and about how sex cells, sperm or eggs, are created. So they wanted to do a sperm cell, and when I showed them pictures on Google Images of different sperm cells taken with electron microscopes, they found the one that had 2 tails. It was a deformed sperm cell, and they decided they wanted to make a 2-tailed deformed sperm cell found object art. It was pretty funny. So as you can see, we're starting to incorporate some of these other more traditional topics from a biology class into this. So they're thinking, they're learning about all the different organelles of a eukaryotic cell and what those organelles do. So they're starting to learn about cellular respiration, for example, is taking place in the mitochondrion. And, they're learning all these concepts that often are kind of the dreaded concepts for students in a non-majors biology class. We had students bring in all sorts of interesting things, whatever they wanted. One student actually brought in this interesting cat basket to be a nucleus, because its head comes off and then you can see a nucleolus inside. We ended up not using that because she didn't want to mess up her cat. But they found other things, like she brought in a piece of a train track to represent part of the cytoskeleton, which serves as kind of the railroad that organelles move along. And then they went out on campus and got branches and worked together to form the cell membrane of this cell. Here are the 2 tails of the sperm cell. They were hanging this thing from the ceiling. They ended up, for the nucleus, they actually ended up using one of those Halloween buckets. They did all these very strange objects that they used their creativity to say, okay, how could this represent a particular organelle and or its function, and how can we put this whole cell together in an abstract art form? And so you can see this is the picture that inspired them, the 2-tailed sperm cell. And this is their final product here. So this this object here, which is still hanging in the lab right now, is the sperm cell. So you can see they used bubble wrap as to represent the cell membrane, phospholipids. They use these what they call noodles to represent the flagella on the cell. Here's the nucleus. They recycled some of the components from previous labs to represent certain things. Here she's using fruit roll-ups and other things to represent a Golgi apparatus. So again, Val and I were hands off on this. They did this all on their own, and it was really, really interesting. And I see no reason why this couldn't be in a museum just like any other piece of abstract art. And so you can see they had a lot of fun with this. And what was really great is seeing them work as a team and think out of the box. I think it was a great example of watching our students grow over the course of a term. And last but not least, their final project was they were asked to create what's called a zoom-in photo book. And the idea is we wanted to take these concepts they had learned the whole term about how life is beautiful at multiple levels and show this in an art project. And so they brought back their skills with using a compound microscope and a dissecting scope. And then they also had to go meet with our own Murray Gans to learn how to use the electron microscope. And so what they were supposed to do, because we also wanted to incorporate photosynthesis in this, is they had to go out and get a piece of a plant. And they could also do an insect if they wanted. But they had, for example, they would go out and get a leaf or a flower, and they'd come back in the lab. They would use the dissecting scope to show what this object looks like under a dissecting scope, where you can still see it with the macroscopic eye. And and they also took pictures with their camera or cell phone so they can see what that looks like to the naked eye. They used the dissecting scope and took pictures. Then the compound scope, they prepared wet mounts of the same tissue. And then finally, they took the same tissue and looked at it under the electron scope, microscope at different levels. And so from all these pictures they were taking, they created a book that would zoom in from the macroscopic all the way to the ultramicroscopic to kind of show the beauty of these specimens at all levels. And they also had to use Photoshop to alter these pictures graphically. So they're learning Photoshop and graphics art at the same time as understanding things like proportion, scale, photography, form, and displaying this into a book. They were supposed to actually get their pictures mounted into a really nice book at, they could use Walmart or send it off, but it would give them a keepsake that they would be able to look at, for years to come. So they're learning many different biological concepts as well as art concepts. So we're featuring two of our students' books here. This is Larry, so you remember Larry, and here's Murray that he worked with. Here's an electron microscope, a compound scope, and a dissecting scope. And here, of course, is Photoshop. And so you can see he actually did both a plant as well as an ant. And here's his final book. This is a leg of the ant. He used Photoshop to make it red and to alter kind of the shape a little bit. And you can see that all these photographs were taken by him. He did all this, the zooming in. Here's one of the ant's eyes. And so he's putting these together in this book. And it was really, really, it was a work of art. It was really, really wonderful what he did there. And hopefully you'll be able to see this being passed around. And here's Xandra, and she did this leaf right here. And so she took all these pictures. Here's with the dissecting scope. Here's the compound scope, so you can actually see it. She zoomed in on the stomata of the leaf. And then finally with the electron microscope, showing the detail there. So very, very cool how she put all this together. Now, people always ask, okay, how do we know if our students are learning? So if the pictures themselves didn't tell you, we actually have a little bit of quantitative data that we're hoping to build on in future years. I think the best way of assessing if our students are really learning is qualitative. You saw those pictures, you saw their faces, you see the way they interact with each other. As faculty, we know when our students are learning because it's like we can see the light bulb go ding, but we like to have quantitative statistics too. So we started with 11 students in this class. So that's 11 for 1301 art and biology 1408. And we ended with 11. So we had 100% completion rate and all of them passed the class. So we had 100% success rate. And I can tell you, I don't have the numbers in front of me, but just personally, compared to my traditional biology 1408, That is a much higher completion and success rate than I get in a traditional classroom. So at least preliminarily with a small sample size, it seems to me that doing this kind of integrative team taught learning community style, project-based, student-driven, I think it really is improving the overall success and learning and long-term learning and critical thinking skills that we want our students to have so badly. Thank you so much for joining us. Please let us know if you have any questions. We have tons of people to thank for allowing this class to go and be successful. We couldn't have done it without them. And I'm sure I've accidentally left people off this list, but thank you for everyone who's helped us and all of you who support learning communities at Eastfield College.